Salut à tous. Oui, ça fait longtemps. Et non, je ne suis pas mort.
Toujours vivant.
Ça fait longtemps que j'ai pas sorti de vidéo et celle-là a été plutôt longue à produire. Dans la vidéo qui va suivre, vous allez voir une interview que j'ai réalisée avec deux paléontologues brésiliens qui sont des amis, Heitor Francischini et Voltaire Neto Paes. Ils sont tous les deux spécialisés dans une branche bien particulière de la paléontologie, vous allez voir, ou sinon vous avez lu le titre de la vidéo en fait, tout simplement. Comme je vous le disais, c'est deux amis que j'ai eu la chance de rencontrer quand je suis parti faire un stage au Brésil en 2016. C'était pour valider ma licence.
Ah, c'est trop bien.
On va parler de leur parcours, on va parler de leur métier, on va voir aussi un peu une dimension humaine de la paléontologie, vous allez voir tout ça. Je me tais, j'en dis pas plus, et je vous souhaite un bon visionnage.
So, Heitor Voltaire, as I said, I will introduce you with a little passage that I will put in the montage at the beginning of the video. And I think it can be interesting to know what's your scholar and your university career. Did you study more biological way or geological way? Which one want to begin?
Okay, so I am biologist.
Okay.
I have my degree on biology, and I did my PhD, my master's and my PhD on geoscience. So I have both the technologies, and actually I partially completed my PhD on the New Mexico Museum of Natural History and Science in New Mexico.
Okay.
So I have some experience in another institution, but yet in geosciences.
Okay. And you, Voltaire?
So yeah, I think our paths are similar with Heitor. I did all of this in the same university, so I am a biologist, like Heitor. I did the bachelor perspective. We can choose between to be a teacher or a researcher. That's the distinction. And I followed the bachelor, but I tried to complete the teacher side, but eventually not finish it yet. I will try to finish that.
Okay.
But then I do my master's, where I met Heitor. We did in the same institution, which is in the Federal University of Rio Grande do Sul State, in the Geoscience Institute, in the postgraduate program. And we'll talk about later, but that's the part of my life I dedicated to the technology. But yeah, my PhD was also in the same program. And like Heitor, I did part of it in the United States, like in Arizona, in the Petrified Forest National Park. And it was co-advised by Julio de Zorro from Argentina, and the United States was supervised by William Parker. And it was advised here in Brazil by Marinho, which was at the time.
So you two, you experiment to study in other countries, and I said that because for the people that will watch the video, I think it will have, for example, few studies, few students, sorry, and it's important, I think, because in France, experience in other country is very researched by researchers, for example, for thesis, and it's a plus point for masters. So it's interesting that in the other side of the Atlantic Ocean, you also have the way to explore with internship and after your study, I suppose. the paleontology in other countries. So it's what your career, and now we will talk about today. What's your current job? Your topic of research? Did you only research, or did you also? I remember when I was in 216, you prepare fossile, Voltaire. Did you continue to do that?
Okay, so for now I am assistant professor at the Universidade Federal do Rio Grande do Sul, as Voltaire said before, which is based in Pörto Alegre city, which is the capital, is the main city of the Rio Grande do Sul state, very southern part of Brazil. And in the university, I teach classes for the undergrad courses of biology and geology mainly, but also to geography. And I'm associated to the postgrad program of geoscience. So I advised some students there too. Actually, my research issue, my main issue in paleontology is ichnology, with a special interest in fossil footprints, and I'm trying to relate some preservational forms to different environments, especially desert ones like dunes, interdunes, sand sheets, etc. But I'm also advising several students, undergrad and postgrad students, on coprolites, that are fossil dungs, and bioerosion. So I'm kind of studying several issues inside the ichnology at the same times, and indifferently to the geological time and to the taxon of the producer.
Okay. And you, Voltaire?
So yeah, after my PhD, it was a brief period without achieving a research perspective. Like
you researched postdoctorate, so at this moment?
Yeah, so when I finished my PhD, I spent some time doing, I don't know how to say it in English, but
a time to think what you want and...
Not exactly that. I tried to apply for many postdoc projects and also for other positions.
Okay.
But for some times I worked with a company.
Okay.
Construction company, and found like monitoring.
Yeah.
The sites where they dig to see if they found fossils. And for one period it was that. But nowadays I am at São Gabriel city, which is like 4.5 hours from Pörto Alegre.
Yeah.
It's in the same state where Heitor is, but not in the same city. So
I will put a map to.
Yeah, that's better because we are in the bottom end of Brazil, like in the south, and here is where you found. We will talk about this probably, but is the single part in Brazil where you found Triassic vertebrate fossils. Yet, maybe another part of Brazil have Triassic with fossils, but we don't know. But that's the single, it's a very straight very straight part of Brazil in Rio Grande do Sul state where the Triassic flourish. But I am not in that part. This is the center part of Rio Grande do Sul. But São Gabriel city is a little bit south to this Triassic layer, and we are stratigraphically.
Discontinuity?
Lower.
Ah,
yeah,
okay.
We are on top of Permian rocks.
Okay, you're near the Permo-Triassic limit.
Yeah, so we are at the top of the Permo-Triassic boundary, and that's actually what I am doing here. So I'm now a postdoc student, a postdoc.
Which animals you study in this rock near of the limit of Permian Triassic?
That's the thing. So this is a project. I don't know, two-part project of Harvard University and Unipampa University. So here, where I am now, is the Unipampa, that is the Federal University of Pampa. So Pampa is the ecosystem we have in Argentina, Uruguay, and in part of Rio Grande do Sul.
It's the area of gauchos.
Yeah, where the gauchos live. That's the idea. And so the project is pretty nice. So the project is to research the Triassic, the Permo-Triassic boundary, to see what happened with the vertebrate fauna. So we have a lot of fossils from the latest Permian, which it's not exactly the extinction event, but it's almost there. And you have early Triassic fauna.
You compare this with because in Brazil it's the Paraná Basin, if I could remember, and so maybe you can compare with the boundaries in the Karoo Basin, because it's the same one at the beginning, no? It's
so that's the idea.
Okay.
I'm now working with these vertebrates, like they are mostly terrestrial tetrapods, but also aquatic ones like Temnospondyls. So we have some of them also here. But I am one of the positions is like a classical paleontologist, which is my position. But we have also Arielle Machado, that is also a postdoc student, and she works with the statistical part of this. So she's building up a huge database for compare these associations.
That's so cool.
And just to mention, like this project was built by Stephanie Pierce in Harvard and Felipe Pinheiro here in Unipampa.
So now we people that watch the video know better who you are on the topic and your work. We will speak about the big topic of the video, the ichnology, because it's with the ichnology that I met you in 2016 when I was a young student. I remember I compare Tucutucu's cat with the Cynodont, maybe scat, or maybe other things, we don't know. And it's in this context that I met you, you and your advice. And so when I begin to thought about realize a video about ichnology, I said me, who is the better to speak about ichnology than the two one who teach me ichnology? So you are. I think the beginning, the better, it's to present in the first time what is ichnology.
So I can start, but Voltaire will help me. So ichnology is a science that aims to study trace fossils, basically. So trace fossils are traces of the activities of the individuals when they were alive.
Okay.
And as example, as a matter of examples, we can use fossil footprints or actual footprints of extant animals too, burrows or paleo burrows. We can use feces, dungs, cats, or fossil feces that are called coprolites. Anyway, any vestigium of the activity of an individual, of a organism, that is preserved in a substrate.
Okay.
And this substrate can be part of another individual, or can be inorganic matter. It's a very huge science.
if we go with this definition, I have the question of the approach of the ichnofossile. So you have you have a biological and a geological formation. So when you have your ichnofossile to study, you use a biological approach. For example, you have a scat or a footprint. You will say, okay, I will watch on the current animals and I will compare. Or you use more geological approach, context of fossilization, the mineralogy, etc. Or you use maybe the two.
When Heitor was talking, he mentioned that we can use the extant traces and also to see the past. So these are almost answer your question. We use the extant world to try to understand the past. And I think that the most striking or most interesting part of ichnology is that it preserves the behavior. So if you want to understand behavior, you eventually will use the knowledge that ichnology provides, because it's the direct record of behavior is only found in ichnology. So for a biological perspective, which is usually what drives people to paleontology, for example, behavior is ichnology, and that's one thing. But I think Heitor probably have a huge knowledge about the geological perspective. So you have a lot of information also for the paleo environment, for example, relative to ichnofossils, like paleo soils. Also have a lot of traces preserved within, so you can use these kind of layers that have these different ichnofossils to understand the paleo environment and use it for stratigraphical purpose, for example. So you have the two strategies, but also, if you want to see the past, if you want to have an idea of what was the Cretaceous, you can use the vertebrate plants, invertebrates, but you also can use ichnofossils to see the big picture of the environment. So this is one thing.
Okay.
Yeah, I will add the information that this answer depends on the ichnofossils, or depends on the trace fossil you are studying.
Okay.
So if you are studying coprolites, you using more like biological approaches, or maybe also geochemistry approaches. And if you are studying, don't know, invertebrates, tunnels, you are using more geological, sedimentological, paleoenvironmental approaches. So the very first answer is depending. Inside the ichnology, you can use this very broad approach of both big areas, either the biology, either geology, depending on which or what you are trying to answer. So if I am trying to answer some aspect like behavior, I will use more properly biological approaches, like comparison with modern animals, like morphometry, like morphology of the trace, don't know, the footprint.
Your answer is very cool, because it's the perfect transition with my next question. I thought about ichnology and the difference with the somatofossil, the rest of the body, and I asked me, is it the boundary between somatofossil and ichnofossile is clear and unclear? Because, for example, the cololith, I think it's an ichnofossile, but it's in the body, in the area of the stomach. For the gastrolite, it's the same thing. For the skin print, is it ichnofossile or not? Is it clear or not? I want your what you think about this?
There are too many shades of gray, and some material cannot be correctly assigned to either body fossil or trace fossil. So your example is pretty good. Cololites or gastrolites are not exactly well positioned inside trace fossils, but some people consider that they are trace fossils made of body fossils, just like coprolites.
So at that moment, I was like finishing my grad in biology and starting my master's, I think, or it was the end of my master's probably. And at that time, I was finishing my work with bone bioerosions. So that's I will talk about the full story. I will try to condense it, but let's see. One day I was in the laboratory as a naive student, and probably after I broke something, and Marina, which was my advisor, she mentioned it. Okay, Voltaire, like you broke this, but try to use this material to see what's happening. So she gave me a box full of broken bones, they are almost all broken. So it was not a problem if I broke a little bit further. But the nice thing is that the bones were already broken, so they were preserved that way. They are almost all fragmentary and broken apart. And in some of them, which was the nice part, they have tunnels, like borings, cylindrical ones, and with a different shape. And that was the intriguing part. What caused it? Because at first, some of the borings were not so curved and not so cylindrical. They are just like teeth marks or teeth traces. Like someone have bited the bones and break apart. It was a nice story, but we have these very strange cylindrical tunnels that we didn't know at the time what was.
At this moment, you know it's organic, but you don't know the animals at the origin of the mark.
Yeah.
Okay.
That was not a tooth, like a tooth of a vertebrate could not produce that kind of structure. And we knew that thing was not recent because the fossil was exposed, okay? It could be recent, but when we dig and do the jacket, we bring to the laboratory a lot of pieces that was inside the rock. And these pieces also have this kind of strange tunnels and borings. And so that was the time when I discovered that you have some insects that can produce these kind of structures in bones, of recent ones. So we have a lot of groups of insects that may damage bone when exploring the cacas of vertebrae. So termites and some beetles, like the dermestid beetles, are the most famous ones. But you have a lot of other animals of the insect group that also do that. And also a lot of other invertebrates, like gastropods, can do this also. So, but tunnels apparently is a very characteristic of insects. So we found these insect traces on bones. That's what Heitor is saying. Like you have the fossil of a bone tissue from a vertebrate, but also you have a behavior of an insect that was consuming this carcass of that animal 26 million years ago. So
and you compare with current observation to show it's very similar.
Yeah, I am resuming.
Yeah. You want to speak? You want to speak more in detail just after? Okay.
Yeah, it's
you don't want to spoil.
Crazy than this, but eventually we found a lot of other traces. And Heitor can discuss more, but I think he frozen. But you have also tree roots that can damage bone. Also, you have, of course, you have pathologies also. So you need to consider that. You need to consider. So when you have this structure that was this tunnel, that was like a void hole in the bone tissue, we explore the possibilities, and the possibilities are very, very large. For example, it could be a pathology, for example. So you need to compare it with pathologies that can produce this kind of structures as well. Ichnology it was more expressive in history using marine fossils. So the continental ichnology is more recent in kind of way, and bone as a substrate for trace fossils to be studied is a very, very, very tiny part of this ichnology area of continental settings. And for example, we lack extant studies. So one of the most difficult part was to compare patterns produced by insects in extant bones to those found in the fossils, because when I was doing this research, there was no much papers on it. So
it's very interesting because, as you say, the bones is not anymore the object of the study, but it's the substrate, it's the support for the ichnofossile itself, and it's very interesting to see to see like that. So you have a report of zooming on the object and the bone. Maybe it's not a big one, but at the end it's just like the ground where you will study dinosaur footprints, for example.
No, just to add that vertebrate technology is growing so fast in the last years because people are not just searching for dinosaur footprints, but also searching for another kinds of traces in another kind of substrates. So bioerosion in bones, bioerosion in shells, several kinds of consolidate substrates are growing very fast, coprolites too, and this secondary issue of vertebrate technology is growing up. You mentioned it, Christopher, dinosaur skin impression, right?
Yeah.
In your question. And well, this is a good point because not all the impressions are under the scope of the technology. Like skin impressions are dinosaurs or any animals are not studied by technology because they are not product of the behavior of the activity of the animal during life. So a skin impression can be produced by fossilization processes that are not acting during the animal's life. They are acting after animals is dead. So this is a very, very easy, very nice example. Dinosaur skin impression, dinosaur eggs, they are not trace fossil as well. Well, and then we have too many shades of trace fossils and body fossils like the stromatolites and another invertebrate structures.
So
like nests.
It's funny because you speak about eggs. I learned that when you found the eggs, if you don't have the embryo or if you have just a shell, you give it an ichnotaxa name, but it's not really an ichnotaxa. It's between the two. But it's good. It shows that you have not limited area between the two. It's good to know that.
Actually, the egg names are not ichnotaxa.
Okay.
They are another taxonomy used as ichnotaxonomy,
Okay.
but it's not considered ichnotaxononomy.
Okay,
It's
okay.
another parallel taxonomy, as you can find as example in plants or in
Okay.
stromatolites. They are not considered.
It's another nomenclature. Yeah. Okay.
What we are talking about is that some coprolites, for example, receive it at the time a name, and this name is a genus and a species, ichnogenus and an ichnospecies name for a coprolite. So that's debatable, but we need to see two things. Like for example, the tubes on the not the tubes precisely, but some of the borings produced by insects, at least what I suggested that's produced by insects, received an ichnogenus and an ichnospecies, and these do not mean that we know the producer, the species of the producer, but for a perspective of systematic and science, it's useful to do this kind of systems of nomenclature because it facilitates communication. So when I mention, for example, like cubiculum, Heitor knows in his mind the trace.
Yeah.
At least that's the idea. That's the idea. It can generate a lot of products for its own, and I think coprolites is a nice example because coprolites, everybody knows, it's not it's a huge pattern.
So the name that have the fossil don't reflect the nature of the specimen, but it's more used to know what we talk about, that it's lizard or it's mammals. Okay, it's like give a more simple name than the laboratory name for the specimen.
I think Heitor have an example, like there is footprints that was named like something, and the producer is completely different.
Yeah, actually, before to talk about that, sorry about the dog.
Don't worry.
Before talking about that, actually, ichnology inherited this kind of structure of description from the botany, the botany, because the very first trace fossils were considered a fossil algae. And then these algae were described and received some generic and specific names 2 centuries ago. And as ichnology grows and grows, it inherited this kind of system of classification. So it's not the very best way of classifying some non-biological structure, but is historically used. We follow the same rules of the zoological nomenclature code. So we have the rule of the priority. So if I name it something, it is the correct name forever. So in the past, some researchers named it, don't know, a footprint called Dicinodontypus or Procolophonychium, and they are now used forever. But today we don't understand these footprints as derived from Dicinodonts or Procolophonids.
Okay.
So it's kind of weird, but is still the very best way to communicate.
Okay. Few minutes ago, you saw in my mind, because you explained that the last years the ichnology developed on even today. So I arrive at my question. So is the ethnologist community developed also, or is it the same researcher that produce more and more data, or is it the community of ethnologists grow? And also in my question, is it in some country, specific countries, or is it a researcher in ethnology community all around the world?
Depends on the taxa or the group of trace fossils.
Okay.
So one of the main important community of coprolite researchers are in Poland and Sweden.
Okay.
And then in the United States and South America. But there's no too much bioerosion researchers on these countries. Like,
okay,
perhaps we have more guys studying bioerosion in Spain and Portugal, in South America and Africa than in Europe and in the rest of Europe.
Okay. We arrive at the part. I think you wait like this, Voltaire. I saw you. The part where you will explain your story. You will explain a few papers, few studies about technology that can be a part of you, or are very interested by the approach. Or literally, I wrote on my paper badass example of ichnofossil studies about that.
I think if Heitor want to talk about the other one.
This is a very nice paper because this changed kind of too much from the very first time to the submission and from the acceptance of the paper. And well, the best part of this paper is that Voltaire and me, we shared too much information and friendship, and this is the best part. But well, this is a paper that was published in 2016, I think. The paper is about materials from Cretaceous, from late Cretaceous, so neither Voltaire, neither me, we were used to work with these rocks, so it was challenging. And then we have some materials that remember coprolites, and in the beginning we thought we were working with coprolites really, and then we tried to study these coprolites, especially because some of them present and some trace fossils on the surface. So it was kind of astonishing because we have coprolites, we have trace fossils, so trace fossils on trace fossils, and then we were searching for some explanation. So what kind of animal, if there was an animal that made these traces? So we start to try several approaches to better understand this material, and our first hypothesis was that these structures, these trace fossils on the surfaces of the coprolites were made by flies during
the larva?
pupation.
Yeah,
by the larva during the pupation moment. So they searched for the dung and then burrowed a chamber and then pupated. So we were pretty happy with this,
Yeah, so like dinosaur dung with interaction with insects. Whoa, crazy.
Yeah, with flies, populations. And we were pretty happy. But on the other hand, we were also studying the coprolites because we tried to understand which animal produced that dropping, that dung. And for late Cretaceous, we have huge sauropods like the titanosaurs. We have several theropods lineages. We have another large animals like crocodiles and so on. So, which animal was the producer of that dung? And then we used a very geological approach, that is the petrological thin section. And in this thin section, we found nothing that could prove that those structures were coprolites. So we were kind of jaw-dropping.
So they failed the leak to identify the coprolite. If someone is seeing, if you found something in the street that resembles a coprolite, you can put your tongue on it and it will be stuck. So that's a coprolite. And those failed on this.
You don't do that for current animals.
No. Just coprolites, not recent feces. That's this.
Yeah, it was very strange because in any moment the coprolites had a coprolite shape. So we were kind of too much help for nothing. Well, and then all the petrological sections indicated that perhaps we were not leading with coprolites, but to lithoclasts, to perhaps sedimentary lithoclasts. Of course, they were sedimentary. They were not igneous clasts, but sedimentary, and they present some aspects of paleosoil structure, like some minerals, some I forget the name. Some
clays, maybe.
There were some clays, but I am kind of remember. Well, the face of the rock, the structure of the rock were too much rocky, and too very to feel coprolite.
So an abiotic origin for the object, and not biotic produce.
Probably not. We don't found any bone remain. We don't found any plant remain. We don't found any microscope remain like pollen or algae, whatever. And we don't found any fabric. This is the word I was trying to remember. We don't found any fabric that can indicate us that those materials were coprolites. So we turned the table. So for now on,
all of this is on Heitor. Heitor have this
I led
the
coprolite part of the study, and Voltaire led the bioerosion or biopreservation.
Because the other traces remain as a trace, but now nothing a coprolite anymore, like now lithoclast.
So you arrive at the conclusion of the coprolite is not a coprolite, but which for the trace.
Yeah.
It say the origin of trace that you can explore.
But it was not a fly anymore, and it need to be an animal that can burrow on a very hard substrate, because that thing was always that shape. At least the substrate not in that shape, but it was hard, and it was always hard. So it was definitely not a larvae of a fly.
So lithophagous animals, like some snail
Not
that
necessarily.
eat rock.
Not necessarily lithophage animals, because we don't know if the animal really eat the sediment.
Okay.
So animals that were, or organisms or individuals, that species that were able to bore to hard substrates. So we start to compare to another animals that were able to bore hard substrates, but they are mainly restricted to marine areas. So we found bivalves, so we found sea urchins, so we found, well, too much animals in the marine realm.
And this fossile is in a continental context. Okay.
Yeah.
So that's the
So
problem.
in the continental context, we are seeing nothing to help us to understand these traces, but they were identical to the marine ones. So
yeah, it's literally identical. It was surprisingly identical.
And then we start to get crazy because we tried to publish that, and this was the very worst part of the job that we facing with the editors and with the reviewers, and then kind of cut some information of our paper because the paper need to fit in some science area supposedly, and we give this more broad hypothesis that there was some kind of individual. We are not sure if they were insects, if they were bivalves, if they were fungi, or whatever. But there are some individuals that were boring structures in the late Cretaceous, in a continental realm. For now, we don't have some prospects like any other paper around the world could help us with this issue, but perhaps we are now more skeptical about flies and dungs and more open-minded.
We think twice prior, like accepting an hypothesis as a
Yeah. So, well, in the end, we named this trace fossil as Astenopodicnium phallax. Astenopodicnium is an echinogenius that were known for a very long time. So we placed our trace in an echinogenius that is known so far.
Okay.
But we erected a new echinuspecies, phallax, Astenopodicnium phallax, and phallax just meaning, I don't know the word in English, Voltaire. Remember me. Deceptive, I think.
Oh, okay.
Yeah.
So
Okay. So all this study was a fight against destiny and sometimes deception, but it's cool because it's also a case for I don't know, the student that will watch the video, that you can go in your research with an idea. You say, I'm sure it's this, and you step by step you arrive at the conclusion of it's not, and you should to pass the deception to find other things, and at the end you arrive at the conclusion that we don't know a lot of things, but the few we know it's a lot. It's not a revolution in paleontology, but it gives perspective for the future, and the history that you tell, it's very interesting for that. I think.
Yeah. This paper, this research was very interesting. Like I remember Agustin Martinelli, which is another collaborator on this research. He's another paleontologist from Argentina. I think he saw for the first time these strange traces and bring us the idea to work with that. And I remember him saying that it was the most amazing research. Like for a scientific perspective, it changes. And through science, like you see science advancing in the research. It was very, very interesting, and I think it expanded our views of how science works, like how paleontology works, and how new technologies can not new, but how different multidisciplinary techniques and a lot of people can collaborate to address a problem. Not that we manage all of the problem, but we can at least say something.
And it's very interesting because if you say that you see the same shape of ichthyophossil but in marine context, it seems that maybe you have like Lazar taxa in fresh water. We don't have the fossil of it, but we know they are here because it looks like what we have in marine deposit. Or if it's not the same family group, maybe the same behavior. So in the two cases, interesting because you have the same thing, the same shape in two different contexts. So in one case it can be the common origin, biological origin, or in another it's convergence of behavior. But in the two cases, it gives two future axes of research very interesting to try to know more about that. So that's very, very.
This is the bad part of the work. We found this perspective during the paper writing phase, but during the submission, the editor asked us to change some things, and then this was the cutted part. But surprisingly or not, more recently, like 2018, 19, we don't know, an English student or an English researcher published a very big review of bioerosion, and she considered our trace as the same as the marine ones.
Yeah, like we mentioned it in the earlier drafts of the manuscript.
So this information was cut by the editor.
Yeah, but
so
now
we lost this information, but now this information is the more accurate because an English said that.
The complex word of article parution.
Well, as Voltaire said, this paper is about scientific method, not about just technology. And perhaps the main phrase that we need to remember of this paper is that not all the shit are shit. Perhaps they can be just a piece of rock.
It's a very good, a very good simplification of all of that.
But as you would say, believe in your word, continue, but not every shit is shit.
But our paper was about bioerosion, but it was not boring.
Yeah,
it was not and it's the most important. Do you want to present a second one? We have a little
I talked too much.
Just to mention the second paper that was from my master's. And that's the story I was begin to talk. So that one's the finished, not finished, but part of my work with the insect bone borings. It all began with that particular one with the tunnels and fragmentary bones that I mentioned earlier. But eventually we discovered a lot of other bones that have this kind of weird, not tooth in origin traces, and that could be insect ones, but also could be pathological ones or other stuff as well. So we eventually found that there was a brief mentioning that in 1999, I think, if I'm not wrong, there was an abstract of a congress addressing insect borings in Dicynodont humerus from Brazil. And this is the single Triassic interaction between carcass, vertebrates, and insect. And it was an abstract of a congress. So we contacted the authors and invited them to collaborate. And then we discovered that this is just one of the many bones that that locality have this kind of traces.
It's a bone bed in reality. You have a bone bed.
Yeah, you can say that. I think Heitor, it's a bone bed.
I don't know. It's not a bone bed, but a taphonomic accumulation.
Yeah, it's a taxonomic accumulation. So it's pretty mass material, well preserved. It had the peculiarities of the middle late Triassic that is like a little bit deformed and a little bit expanded sometimes. But you can see the traces very well on this sample. And we joined together and searched for more bones of the same age, and they're still the oldest one published so far. Like, we have these middle late Triassic bones with some vertebrates like Dicynodonts. I think they are mostly Dicynodonts. And then you have a gap, and I think Heitor is researching on these gaps. But you have like sorry, no spoilers. But you have like in more recent strata as well. We have like the other kind of traces of insects, and those are very, very nice. You can see they have tunnels, borings, and pits, for example, that they're most common ones. But you can also see the mandibles, like the mandible scratches, like they call this striae. You can see them, and I'm not giving spoilers, but but we published the first ones, and they are not so good as the future ones. Oh, sorry, it's a spoiler again.
We are using the Voltaire's paper to pave a new road and searching for new traces.
Okay.
Amazing new stuff will come soon.
Hope that is soon.
I think we speak a lot about technology. We give a lot of example for people that watch the video. So I hope they will enjoy it. It's not an happy subject, but I think it's important to speak about. It's for the case of the Museu Nacional do Brasil, when it was on fire in 218, and because even in France we hear about that, and more precisely in the university and paleontological community, and I wanted to know today what is the state of the reconstruction, but not only the wall, the building, also a state of knowledge, what we lost, what we could preserve, how it could impact the paleontological community. I just wanted to know your what you think about, what maybe you can say.
Well, actually, the answer is we don't know. work is still in progress, and what we actually know that too many specimens of another collections that were not paleontology were completely destroyed, like the anthropology and the archaeological collections, the entomological collections. They were very, very damaged. But about paleontology, we don't know. We have our fingers crossed. So we wait that the damages can be minimal, but they were very severe. The progress on the reconstruction and on the recovering of the material on the building is very slow because there are too many archaeologists working on that, so they were working very slowly to recover all the pieces with care and so on.
It will take a lot of time to reconstruct and arrive at the state it was before the accident.
I think it is scheduled to be open again in 2017. No, 2027.
Okay.
There is a plan to recover part of the material, recover part of the collections. There is a schedule named Recompõe, that means rebuild, and the current director of the museum are talking with several another museums in Europe, in the United States, in another country, to try to have some material to the new exhibition.
To reassemble a collection to exhibit to visitors.
Yeah, for those who don't know, the National Museum was for Brazil, I think it's the oldest scientific, naturalist, anthropological museum, and it was amazing. And it was part of the history of Brazil, the building.
I had the chance to visit it when I came, and it's something to visit if you have the occasion, because it's not only natural history, it's all the South America history and Brazil history. So yeah, it's a great loss.
Dom Pedro II, the second was the emperor that was an enthusiast on nature and anthropology and history. So he collected, when he was alive, a lot of materials, and he started this collection. And it was building for like, I don't know how many centuries. And yeah, it's all burned, and it was very sad. But as Heitor said, for the paleontological, the vertebrate paleontological, I think we have some promising news. Like some of the material is still there, but eventually not entirely preserved, but part can be recovered.
All is not lost. Okay.
Yeah, something will come from the ashes.
That's good.
And recently, a dinosaur was published. One of the dinosaurs, I think it was on the museum.
But we faced also another problem that is as severe as this kind of problem, that is the fossil traffic. So we also have some problems with material going out of Brazil, especially to Japan, Europe, and United States. So perhaps your public want to know that material fossils from Brazil are considered national heritage material, so they cannot be trade, neither collected without permission. and we are fighting against some with another country rules that are considering still receiving Brazilian materials illegally. Yeah.
Few countries are more strict than another, but for example, Morocco pass a lot of fossile. But I saw that I know that other countries like Brazil, no one passed without permission. But it's a good thing because, for example, in this case of the fire of the museum, delete this traffic is also a good way to recreate collection to exhibit for visitors and materials for researchers.
Yeah, we need to some kind of collaboration between especially the European countries that are the most receptors of this kind of material, especially France, Germany, and England, United Kingdom, but also Portugal, Spain, I don't know, all the countries. And the governors need to contribute to the Brazilian government to this, to trade them back, essentially, because
the main thing is that these fossils that go outside Brazil goes out illegally, so they lost the context, they lost because they are not digged by specialists mostly, and we lost completely our context. So when they are deposited outside Brazil illegally, because there is legal ways to have a Brazilian fossil in your institution in Europe, for example. But usually that's not the case. Usually they were bought for a trade, a legal trade that goes to Europe, Japan, or United States. And I think this is a common problem in America, including in the United States, for example. But also Argentina suffers from the same problems. In Brazil, especially those materials from the northeast part of Brazil, from the Cretaceous, like Anguera that you mentioned. And most of, I think that's the paper recently, most of the plant fossils are outside Brazil. So yeah, so imagine that if you are Brazilian and live on the Ceará state where this fossil came, and if you want to study an insect, almost all of them are outside Brazil, and they were eventually and all illegally trade, not legally. That's the problem.
It's in the same way of few countries that, for example, come back artifact or archaeological rest. I think, for example, at the Maori of African countries, it's in the same reasonment. To reappropriate your own history, natural or human history, but it's the same. Okay.
Exactly. Yeah, but the thing is, this happened recently, like the Strait. It's not so historically in the past.
Yeah, it's not a colonial history, or it's just it's recent and we come. Yeah.
It's a colonial derivative, but it's not that much in the past.
Yeah.
It's recent.
Okay. Do you want to add something before we end, or because we switch little by little on the fossil traffic? It's also a good topic of discussion. If you want to add something, or maybe on another topic,
we are missing you. We are waiting you for a visit.
Yeah,
and this is the most important.
Yeah, it will be with pleasure. We organize that, and the reverse thing. It's also good. You have a little part of Brazil that waiting you in France.
Bon, alors j'ai coupé ici parce que après on commençait à parler de trucs qui touchaient plus à la vie privée, qui étaient un peu moins intéressants pour vous. Promis, je mettrai moins de temps pour sortir la prochaine. On se retrouve bientôt. Je vous souhaite de bonnes fêtes de fin d'année. C'était Christo. Salut.